All right. So I'm going to go back to the beginning just for the benefit of the recording. So today I'm talking about pitfalls in microbiome accuracy and reproducibility. So these are some common problems that are coming up. And I remember this from when I first, from even before I was working in microbiome. I think I heard about it first in on an NPR report. But there were two major studies, sort of a citizen science projects that were happening. There was the Human Microbiome Project, and this was based in the U.S. I should say it is based in the U.S. because it's still ongoing. And there's MetaHit, which is short for the metagenome of the human intestinal tract. This is based in Europe. So HMP was primarily testing Americans. MetaHit was primarily testing Europeans. And they reported some big differences in gut microbiome composition. So it's pretty well known that in a at least human stool sample and most other stool samples, your predominant organisms or your predominant phyla are going to be firmicutes and bacteroidetes. Firmicutes are gram positive, bacteroidetes are gram negative, and they are and there's some pretty big biophysical differences between them that aren't always completely appreciated by people who are doing these studies. What was interesting about this was everybody was postulating different reasons, and a lot of people were saying it's due to differences in the American diet versus the European diet, or lifestyle differences that are not diet related, just a bunch of different possibilities. And it actually turns out that these two differences could be explained entirely by technical differences. So you could actually explain this difference just by how these different studies were extracting microbiome DNA. The reason for this is microbiome is actually a fairly complicated pipeline, and even the individual steps tend to be complicated. So sample collection and preservation, that's actually not trivial. Microbiome is an extremely perishable sample, and until you actually kill it or stabilize it, it's alive. And bacteria are constantly undergoing selection, so once you remove a microbiome, it's going to start undergoing different selection than in its normal environment. You have your DNA and RNA extraction. That actually turns out to also be a big deal because, as I mentioned, different microbes have very different biophysical properties. Library preparation. So we've gotten much better library preparation, but you can still run into issues such as using suboptimal primers for targeted sequence. Some library prep methods, although I think this has been addressed a lot more recently, have issues with GC bias, so you tend to miss organisms with either high or low GC content. Sequencing, I consider this to be more or less a solved problem. Every sequencing platform does have its own little quirks, but most of these things are figured out now, and they're pretty well mathematically modeled, and a lot of times they aren't creating huge amounts of bias, or if they are, the biases are very well understood. And then bioinformatics. And as it turns out, bioinformatics for microbiome, that's still kind of a that's a pretty open field. There's still a lot of things that need to be solved. And just to be clear, when I talk about bias, bias is what you have when you have a systematic error that's pushing your results one way or the other. So as I mentioned, I think on the first day, just randomly dropping out 10% of my reads from a sequencing study, that's not a biasing error. That's going to cause other problems. So that's going to cause me things like rare samples or rare sequences. I'm probably going to lose detection on some of them. I'm going to lose statistical power. So smaller effect sizes or more unusual things, I'm probably going to have trouble detecting, whereas previously I wouldn't have. But it's not going to necessarily change my my results in the end. There's probably going to be some things that previously I could call that I'm now going to say are inconclusive. And there's probably going to be, there's probably going to be some things that I could call before that I could say are inconclusive. And there's going to be some things where previously I had high confidence that are now going to move into moderate to low confidence. If, on the other hand, I drop out 10% of my reads, but I drop out the 10% of reads with the highest GC content, that's incredibly biasing because I've actually started removing very specific things. And there's probably going to be some things that just disappear entirely. So as I mentioned before, there's going to be one Zymo product that I mention. I generally try and avoid getting too much into brand names or specific products because obviously I have a little bit of a conflict of interest. But one of the things we sequenced was or one of the things we worked on sequencing for is the Zymo-Biomics mock microbial community. This is actually what it looks like. So if you want to take the data that I gave you and you want to just see how it looks like compared to this, it's actually fairly easy to do. You just have to do composition tables and that kind of thing. But what we're looking at here is we have a bunch of different microbes. If you notice the two at the bottom, these aren't bacteria, these are actually yeast. So we have gram-positives, and for those who don't remember their Microbiome 101 class, gram-positives are primarily defined by having a single membrane and a very thick peptidoglycan cell wall. So relatively physically tough cells. Gram-negatives have a thin cell wall that's got two membranes on it. So these are held together mostly by membranes, and membranes tend to be soft and they fall apart easily. At the bottom, we have fungus, so specifically yeast. Fungi tend to have cell walls, and what's interesting about them is their cell walls are made of chitin. And if you're unfamiliar with chitin, chitin, among other things, is what makes lobster shells. So chitin is incredibly tough. And in fact, yeast are some of the hardest organisms to lyse out there, with Cryptococcus neoformans being incredibly physically resilient against things like boiling and all these other things. Now, if I boil Cryptococcus, it will die, but it won't lyse. So how to measure accuracy relative to a standard? And this is based off of, a lot of this is based off of work that's not yet published, but I'm actually, in fact, once I'm done teaching, I'm going back to writing up this paper. And this is a technique I developed. It's a bioinformatic technique. And you can actually download the Dockers for this and run it on your own data. So measuring accuracy relative to a standard. On the y-axis, we have our observed value. So these are observed relative abundances. On our x-axis, we have our expected relative abundances. And this, again, is from the Zymo-Biomics community standard. And we see here that in a good example, a lot of these dots stay fairly close to this theoretical diagonal, which is we see what we expect to see. In this not so good extraction, we actually see a lot of deviation from there. So we see a few things that are close. We see a lot of things that are actually moved fairly far away from where they should be. And this is just showing Listeria. So I picked Listeria actually because it's one of the tougher bacteria in there. Listeria is gram-positive, and it's just known for having one of the physically toughest cell walls. Now, one of the things we also know about standards is their manufacturing. So one of the reasons I specifically use the Zymo-Biomics standard is the Zymo-Biomics standard actually has not only stated expected relative abundances, it also has stated tolerances. I should say manufacturing tolerances. And these are just based on how accurate they expect to be when manufacturing the standard, given their methodology for counting bacterial cells and that kind of thing. So we know what to expect and we know how accurate we can possibly be with a standard. I do a quick transformation. So now everything has gone from being expected versus observed to % of expected. And we see right along this line, there's 100% of expected. So I think this is probably Listeria again that lands right on the line. So we see exactly what we expect for Listeria. Most of these other microbes, we see very close to 100% in the good extraction. We have our band, we have what I call this band of tolerance again. So this goes 15% either direction, which means it goes up to 115 or down to 85. And the good still looks very good. And in fact, if we look at the good, we realize everything is in or near this band of tolerance. So this band of tolerance again is where it's as accurate as I can say, it's as accurate as the standard will let me say it is, considering its manufacturing tolerance. In the bad extraction, we see that nothing is inside this band of tolerance. Everything is way off. So it kind of becomes obvious at this point how we can quantify accuracy or inaccuracy. We're just looking at these residuals between what we observed in terms of relative percentages and this band where it's as accurate as it can possibly be given the standard. And this is where we have a scoring system called the Mike score. Now, for the record, I did not name this. This was actually named by a colleague of mine, also former UCLA person, who was trying to troll me on an in-and-out run. And basically the response to me saying that I don't want to be the guy, I don't want to be that guy who ends up with I guess, a scoring system that he names after himself. He basically got back to the lab and told, I think, every RA to start calling it the Mike score. So the name kind of stuck, and this is what we end up with. One of the challenges in designing this is we wanted a system that was very easy to understand, even for non-quantitative, non-bioinformatics people. So the system goes with a maximum score of 100. So a perfect score of 100 means there's no deviation from expected values that can't be accounted for by manufacturing tolerance of the standard. A lower Mike score such as 30 means that there's actually quite a bit of deviation that can't be accounted for by manufacturing tolerance of the standard. Or in other words, there's some kind of bias being generated. Although this won't necessarily tell you what kind of bias that is. For that, you need some other information. One of the other things I fairly quickly figured out is these these composition taxa plots that I think everybody's used to seeing from microbiome data. These are pretty much insensitive to inaccuracies. So I can look at this theoretical value, and again, this is just a theoretical for the standard. There isn't really a good way to show manufacturing tolerances, so I kind of lose that ability. But more than that, one organism being way off is going to throw everything else way off. And I can tell the good looks a lot like the theoretical, although, for example, you start to lose the fungus, because the fungus is relatively a smaller proportion. In the actual sample here, I can tell you that it looks pretty different. It's definitely not as good as the good. It's hard to say what happened to the fungus, though. Did they just disappear, or did they just, lose a lot of value? And in this extremely biased one, I can tell that there's even bigger changes to especially these organisms up here. I can also tell you that it's E. coli, Salmonella, and Pseudomonas. So this is massive overrepresentation of the gram negatives, with the gram positives being squeezed, with the gram positives and possibly the yeast being squeezed way down. I came up with the idea of plotting these on a radar plot. And the nice thing with radar plots is I can arrange the radar plot however I want. So I can put the harder to lyse organisms at the top, easier to lyse organisms at the bottom. And we can see here, for example, that this Mike score 93 sample, the data is pretty well centered. So we see that everything is very close to this darkened line, which represents 100% of expected, or observing 100% of our expected value. If something is moved in towards the center, that means we're seeing too little of it, or it's underrepresented. If it starts moving outward, that means it's overrepresented. So something at this line would be seen about 1.5 times expected. Out here, we're seeing double the expected relative abundance of it. Something that we just missed entirely is going to look like a big missing wedge from the pie. So again, this one, it's pretty well centered. This one, it's not well centered anymore. And in fact, what we see now is we have a big shift downward with a piece missing right here. So what I would say about this one is we're heavily biased towards easy to lyse. Not surprising, lysis bias is probably about the most common bias in microbiome research. I also see a dent here, or a big divot taken out. And this missing slice of the pie is at C. neoformans. So I'd say C. neoformans is probably the hardest thing in here to lyse. So Tim is asking, are these OTUs? And Tim is also running into my other biggest problem with the OTU approach, which is autocorrect always thinks you're typing out. And even if I'm typing in all caps where it should be obvious that I'm typing OTU, it still gets autocorrected to out. But I think that's more a problem with autocorrect than OTUs. And it's definitely not the most awkward thing autocorrect has turned something into. Anyway, these are actually ASV based. So I'll show you in a bit, but the pipeline for generating these, it's automated. It runs in Docker. So it's actually very easy to deploy to a computer. And when I show the pipeline, it'll be obvious these are OTUs because these are actually being run by an automated Data 2 pipeline. So Again, Cryptococcus is also missing here. Not surprising because we have a Lysis bias, and Lysis and Cryptococcus has about the hardest shell in the bunch. All right, this is Mike score 30, so this is kind of just an even worse prep with even bigger biases. And what we see here is there's a massive, I tend to refer to this as just a downward shift. So we're actually off the scale for overrepresenting Pseudomonas. We're probably right at about double the E. coli we expected and double the Salmonella we expected. So all the gram negatives are being observed about twice as much as expected or more. Meanwhile, we have contraction up above. So our gram positives are being massively underrepresented. And it's actually pretty clear here what happened because this was an expected pattern of bias. And we can see a very distinctive shift here. So this just gives a really nice data visualization technique for it. As I mentioned before, these are fully automated. And actually, it works for both shotgun sequencing and 16S. So, and actually what I should say is if you're doing 16S, you get OTUs. So in answer to Tim's question, these are neither OTUs nor ASVs. And the reason I can say that for certain is this is not 16S sequencing. Because you see up here that we have Saccharomyces and Cryptococcus. And 16S and 18S sequencing, it's hard to do the quantitative comparison, so a lot of people don't like to. At least between 16S and 18S. This was actually shotgun sequencing. So for shotgun sequencing, you don't have OTUs. You actually just have sequences that get aligned and called. So this was actually just going by raw number of sequences that aligned to each organism specifically. So again, for shotgun, it's a pretty simple pipeline. Start off with a FASTQ pair. Because I'm using specifically the genomes of the expected organisms, I don't have to use any particularly fancy microbiome alignment tools. I'm actually good with just BWA and SAM tools for doing the analysis. I have some Python packages that then interpret the data, generate visualizations through Matplotlib, and they kick out a report that is HTML formatted, so it will actually open up in your web browser for easy reading and visualization, and I think pretty much everyone has a web browser available. There's also a slightly different pipeline for the 16S. So that's going to have the same inputs. It's actually running through the same Python packages as well. This one is using Figaro and Data 2 for the actual sequence analysis and calling taxonomy. And another way to design this is actually not necessarily a Zymo-specific product. Anytime any standard that has known relative expected abundances and well-defined manufacturing tolerances, I can actually build that as a standard for this program in about an hour. It's actually fairly easy for me to do. All right, so let's try and understand why this is happening, because this is actually a critical thing that is in microbiome, and a lot of people don't worry about it as much if they're just doing a single experiment, but comparing between studies, this can become a big issue. And in fact, I think there's potentially going to be a crisis in the field, although to some extent there already is, about the fact that one lab studies and another lab studies, they can't be combined for meta-analysis, and they can't always even be compared to one another. So this is actually data that was generated at Zymo. We were just doing comparison between kits. And we saw some very different results from the same stool sample with different kits, or different protocols, I should say, because one of these is in a kit, one of these is just a protocol. But we saw very different results just at the phylum level. We also saw some pretty big differences in DNA yield between them. And we actually began to wonder if this difference in DNA yield is kind of giving us a hint, because there might have been something left behind. And this kind of gets into a big issue that I think is underappreciated in microbiome, which is gram-negative bacteria and gram-positive bacteria are biophysically very different. So membranes, I think everybody's pretty much familiar with what membranes are like. They're kind of flimsy. They fall apart in detergent, and they also are physically just very soft. what a lot of people don't completely appreciate is peptidoglycan is physically very tough. It's a very hard, very resilient material. Or in other words, gram negatives are kind of more like eggs. Gram positives are more like geodes. The cool stuff is on the inside, but to get to the inside of gram negative, you basically have to expose it to some not even super harsh chemicals or hit it a little bit. To get at the inside of gram positives, you need to kind of whack them with a hammer. And you can kind of see this if you just do a little bit of thought experiment in your head. So E. coli, you use E. coli in the lab, or at least you know of labs that are using E. coli. And people like putting their plasmids in E. coli, and everybody thinks it's just because you put your you can get your plasmid in E. coli easily, and then E. coli grows like crazy, which is true. The other thing that's really nice about E. coli is you actually get your plasmid back out at very high levels. And that's because with E. coli, if you just put them into an alkaline solution with some detergent, they kind of disintegrate. And that would be as compared to something like Listeria, where if you could somehow get your plasmid into Listeria, you'd actually get back only about 50% of the plasmid that grew up, because only about half your Listeria, about the best a lot of people can do is maybe getting half their Listeria lysed with the same method. And this is something that's important to know, because, for example, if I had a super simple microbiome where it was 50% E. coli, 50% Listeria, and I just put the sample into boil for about 10 minutes. So that's gonna lyse all the E. coli, because E. coli tend to fall apart easily, especially under heat conditions. The Listeria are all going to be dead. So nothing's going to be culturable after 10 minutes of boiling. But if I did a microbiome analysis on it, I would actually think it was about 95% E. coli and maybe 5% Listeria, because if the cells don't lyse, then the DNA stays in the cell when you wash out all the cellular debris. And that's actually a very fundamental point with microbiome, is you don't see what you don't actually lyse. So this is more data that was generated at Zymo, and this is actually going to be coming out in a paper fairly soon. I'm working on getting the preprint up actually once class is over. And this is just comparing some different lysis methods. So over here we have bacteria lysing in basically a preservative, and sham metapolyzyme treatment. So metapolyzyme is, I think right now on the market, it's like one of the only ones. It's a cocktail of enzymes that are designed to be bacteriolytic. And what we see here is basically if we don't apply a real lysis method, we get a pretty poor score. And a lot of this is actually just generated by gram negatives, because gram negatives spontaneously lyse. And especially spontaneously lyse if you put them in a preservative, because preservative is not its sort of ideal home. So if we add metapolyzyme, we do see a lot more lysis. That's not surprising. Metapolyzyme is designed to be lytic. So metapolyzyme is actually not as bad as it looks here. It does create some bias. It tends to underlyse a lot of the tougher bacteria. However, metapolyzyme has a property that actually makes it very interesting, which is it doesn't contain DNase. So enzymatic methods, not surprisingly, enzymes have specificity, and specificity is pretty much a really good breeding ground for biases. But enzymatic methods tend to leave DNA intact, whereas physical methods like beads, and I'll talk about those in a bit, actually I'll talk about them at length, beads have a tendency to shear DNA, which isn't a problem if you're doing Illumina sequencing, because your Illumina fragments are small anyway. They're usually on a few hundred bases. But if you're trying to go for nanopore sequencing, where you want like hundreds of thousands of bases if you can get it, beads tend to be a bad option, because they're going to limit your read length. So if we do just proteinase K, surprisingly, proteinase K isn't super helpful as a lysis reagent. It's not actually designed to be one. So yeah, use a metapolyzyme. If we add proteinase K, again, we get a little bit of a improvement in lysis. This is probably just due to clearing out some protein garbage and having more efficient extraction, but really proteinase K doesn't help all that much. If we bead bash after treating with metapolyzyme, we get now mic scores in like the mid to high 90s. So it's actually incredibly accurate. If we bead bash before, we also get very high mic scores, and that's regardless of whether or not we use metapolyzyme. So whether we bead bash before or after metapolyzyme, and again, the metapolyzyme negative is just a sham treatment with the metapolyzyme buffer, we end up getting a very high accuracy with our lysis. And the control is just a sample that we kind of had with a validated method that we could use for monitoring consistency between runs. Thermolysis. So you see at the bottom here, there's a bunch of different thermolysis methods. I'm not going to go through this in detail because, as you probably notice, all these bars are pretty darn low. And what this kind of tells you is thermolysis is not a particularly good lysis method. It's not good on its own, and beads tend to be both necessary and sufficient for the most accurate lysis. So in general, thermolysis is not a method that you should be employing, and definitely not thermolysis alone. Otherwise, you're going to have a very skewed microbiome representation. So we focused on beads, and again, this is more data that was generated that we generated at Zymo. In fact, a lot of these I did the analysis for myself using the mic score system, including this one, which you can see here because the y-axis is the mic score for accuracy. On the x-axis, we have different bead sizes, and what we see here is very different performances for different types of beads. And these are all different beads that people have that are in use right now for different microbiome extraction protocols. So every one of these bead materials is in use and cited in the literature. I can't speak to whether the specific sizes for each material are represented because we just decided to test a wide range of sizes. But what we see here is plain glass beads we get poor performance out of, which isn't too surprising. Glass beads are basically the least dense bead material. And, and Timothy's asking, I thought using beads got mic scores above 90. Why are all of these much lower? Were these not the beads used in the previous figure? That is actually, you pretty much answered your own question there. So these beads are all different materials and different sizes. And clearly in the previous one, especially for the study we were running, we're probably only going to use one type of bead, and it's going to be the beads that we think are sort of the optimal beads. So yes, you're going to see in a moment exactly what we thought was the optimal bead setup, because it's actually on here. So when I talk about the glass beads at different sizes, the glass beads, we kind of figured out early on were not an optimal bead setup. And so those were not the ones represented. So glass doesn't do a very good job. And again, it's because, I think it's probably because of a density issue, although I can't really prove that right now. I'm not sure how I would prove it, but in general, we noticed that lower density beads tend to be less efficient. It's not too surprising because beads work through kinetic energy. And the equation 1/2 mv squared to basically describe the energy that's being imparted is, it's a basic rule of physics. It doesn't change. And it also means that if you have a bead if you have beads of different densities, the denser bead is going to have a much bigger mass. And that bigger mass is going to give you a bigger kinetic energy in the end. So glass, not super performant. If we go up here, we see synthetic garnet, which is more dense than glass and harder than glass. Also, hardness probably makes a difference because if you have harder beads, you have less elastic collisions. So synthetic garnet, slightly better performance than glass. Zirconia silica, which is kind of a glassy ceramic type mixture, about middle of the road performance. We got high performance with zirconia. So either using zirconium oxide, which is kind of the plain version, or zirconia yttria, which tends to have better structural integrity. It tends to resist cracking. We got better performance. We also saw pretty high performance by beads that were about 0.3 millimeters in size. This single dot up here represents zirconia yttria beads of mixed size. And that mixed size is a mixture of 0.1s and 0.5s. So Timothy, to answer your question, this is what was actually used to generate our bead data. So does that answer your question? Okay. So yeah, one of the points of this is beads definitely generate good even lysis. And one of the other lessons I got from this is you really want to have lysis that approaches about 100% of the microbes in a sample, because otherwise, due to the just biophysical differences of the microbes, that less than 100% of lysis, that inefficiency, um, is not spread evenly. And Timothy's asking if these are from Zymo. So again, I'm aside from standards, which I'm just kind of a big believer in, and if somebody else comes up with a standard that also has a very well stated expected abundance and manufacturing tolerance, I will actually discuss that in the class as well. You can definitely get beads similar to this from Zymo. However, Zymo doesn't make beads. The beads are made by a ceramic company. I don't know even where they are, but the beads are actually supplied to us from a ceramic company. And I think they're actually for us. I don't actually even think we make money off them, but I don't know for sure. They don't involve me in the money decisions. They just have me quantifying microbes. But yeah, these beads come from ceramic companies, and basically they get incorporated into kits. We're definitely not the only ones who use this type of bead either. There's other companies that use ceramic beads, although they tend not to be as open with what their bead mixture is as we are. All right, what about bead beaters? So this is something Zymo definitely doesn't make. We do not make bead beaters. So I talk a little bit more freely about different brands on this one, although I'm just showing some examples here. So again, going back, we have a standard, which means we kind of have some expected values to work with. Oh, and also important to note is I classify the organisms for this one to help with really understanding. Gram positives are generally classified as hard to lyse. Gram negatives is easy. Fungus is classified as hard to lyse, and it's actually some of the hardest. And what we see up here is we again have a band of tolerance. So this is kind of the range where we're as accurate as we can possibly be, given the manufacturing tolerance of the standard. And this is 100% of expected. And this is looking at a ratio of hard microbes to easy microbes. And this is using an MPBio Fast Prep 24 for different amounts of lysis time. And what we see here is at 1 minute, we don't have very good lysis. At 3 minutes, we now have pretty good lysis. And we plateau out at a pretty reasonably close to kind of this ideal range after about 3 to 5 minutes. One other thing to note is MPBio Fast Prep is an incredibly energetic machine. And this is the total amount of lysis time. This is not actually the time it would take you to do this lysis. This machine bead beats so vigorously that all the bead collisions generate internal heat in the sample. And you have to turn the machine on for about 45 seconds to a minute and then turn it off for a few minutes to let the samples cool. So this 5 minutes would take closer to 45 minutes on the wall clock. And again, this is our target zone. So we want to basically end up in this range or plateau out in this range if we can, or at least plateau out as close as we can to it. And we see this does a pretty good job. It correlates pretty well to the mic scores. So again, we see at 3 to 5 minutes, we have mic scores basically in the 90s. And this is using that optimal bead mixture. So we're not obviously changing beads around. We're giving it the best possible bead mixture that we found. Vortex Genie plus adapter is an interesting one. So this was also a recommended protocol, but a lot of the recommendations were to run it for 10 minutes, which is what's used for a lot of other bead beaters. Pretty much every lab has a Vortex Genie, and a lot of people are familiar with them. They're not all that energetic. They have fairly small motors, and they run kind of quiet. And what it turns out is if you're running something that's low energy, you just need to run it longer. So something like the Vortex Genie with the horizontal adapter, you start getting good lysis about 80 minutes, and you get very good lysis at about 40 minutes. The nice thing is this run actually just takes about 40 minutes. So Vortex Genie is lower energy, which means less internal heat generation at any given point in time. And in fact, the heat dissipation off the tubes is enough to keep the samples from degrading due to heat or boiling or whatever else. So you basically just set these for 40 minutes and you can actually walk away. If we look at the mic scores again, we get very good mic scores at the 40-minute mark. So how is this affecting the results? Not surprisingly, again, if there's inefficient lysis, if you're not lysing something approaching 100% of your microbes, you can wind up with you wind up with that lack of efficiency being unevenly distributed. So this is actually from a really large study we did on a bunch of different beads, some of which I didn't even show in that chart because we even had metal beads included. But what we see here, each dot represents a different size and bead combination that was run on an identical lysis protocol. And each dot represents at least the triplicate value for each. So each of these dots represents something that was repeated at least 3 times, with the average value being the dot. And on the x-axis, we have a bacteria-only mic score. So that's applying the formula, but that's applying the formula only looking at expected values for bacteria. Because on the y-axis, I have just the firmicutes to Bacteroidetes ratio. So the mic score can only be calculated on a known input sample, which in this case is the Enzymobiomic standard. The firmicutes to Bacteroidetes ratio, this is calculated on stool, and this is looking at the ratio of firmicutes, which are hard to lyse, versus Bacteroides, which are easy to lyse. So if I have a lysis problem, I start expecting a much larger denominator and a smaller numerator. Because the relative abundances are changing because I start missing the firmicutes. And what I see here is a bead method that generated a low mic score. So started to generate, basically generated inefficient lysis. And low mic scores in this case were, I think, universally caused by underrepresentation of Bacteroidetes, by underrepresentation of, or overrepresentation of gram negatives and underrepresentation of gram positives. And what I see here is stuff that scored low for accuracy tended to give me the impression that there was a much higher ratio of Bacteroidetes in the stool. As I start increasing the mic score, so basically more accurate lysis, or more even lysis, or more efficient lysis, whatever you want to refer to it as, I can see a pretty good correlation to increasing firmicutes to Bacteroidetes ratio. So what I start seeing here is that as my method appears to improve, and as I get higher and higher scores with the beads, I start getting better, and I start getting a much different impression of what this sample is made of, which is actually a little bit disturbing if you think about it. This basically means that just swapping out different bead types can give me a very different impression on my sample. The Pearson correlation on this was high. It was like 0.83. Which is especially impressive considering that this is just trying a bunch of different beads. So there's potentially a lot of variability in just how the beads are going to behave, with a very high statistical significance. Let's see, and it's 9:51 right now, so let's actually do a 10-minute or so break. And after we get back, I will go through the final couple slides. But this is actually a pretty good stopping point because we're about to change topics. So we'll pick back up again at 10 o'clock. If you have any questions, I will hang around and answer them. Otherwise, I'll see everybody here in about 9 minutes. All right, I think I can pick up again now. I'm just answering a few questions in chat. And yeah, for Tim, I'm more than happy to discuss this after class. And if changing so one thing to consider with experiments, and this is just in general, although there are some specific kits that I think may be going off the market soon, is try to ascertain if the system you're going to be using is going to remain available for the foreseeable future. And I think Eloy's about to jump in and take attendance. Is that right?
Yes. All right, same drill as yesterday. I'm putting the link in the chat room for our attendance sheet. Please fill it out. After you completed it, just raise your hand. So that way I can keep track of people who haven't filled it out. So yeah, after you finish, raise your hand. So there's gonna be nothing. So I'm looking for 13, no, 12. All right. Should be 12 people. All right, good. All right, thank you.
Mm-hmm.
All right.
All right, so I can pick this back up again. All right, sample collection and preservation. So this is another big issue that comes up a fair amount. So one thing I'll talk about is bioburden. And this, by the way, is another issue with beads that isn't discussed as often, because a lot of times with beads you talk about in terms of material, which definitely material, and a lot of times you can even just break the material down into how dense is it and how hard is it. Another issue with beads is also just how they're prepared. If your beads are covered with biofilm, or if your beads are, somehow passed through a dirty area, your beads are potentially going to have microbes on them. Same general idea for swabs. And I know this was recently a big topic in the news when the president found out that when the president was surprised by the fact that the swabs you need for doing microbiology testing are different than, say, the Q-tips you buy over at Rite Aid. That was kind of entertaining to watch, I'll admit. But and also what's interesting about that is I've seen people during this COVID crisis actually doing some real shenanigans involving swabs, including somebody I know who was trying to obtain appropriate swabs for testing. Somebody offered to sell him some, and when he got a hold of a sample of the lot, he realized these swabs don't look like normal swabs, and then inspected them more closely and realized that somebody basically just took a bunch of mascara appliers, like a bunch of mascara brushes, labeled them as swabs for testing, and marked up the price like I think about 50-fold. Obviously, he did not want those swabs. So anyway, just to give you an idea of what bioburden is and the effects it can have. So here in the middle, we have a typical kind of collection device where it's a swab with a tube. I'll talk about this in a bit, but ideally, if you're using a swab in a tube, you want to put the swab into some kind of preservative to make sure your microbiome doesn't start changing. I'll talk about how that happens in a bit. But these swabs, even if they're marketed as sterile, they may still have microbial DNA on them, even if there's no culturable microbes. But the other thing to remember is if you're looking for microbes by culture, there's this idea of culturable but viable but not culturable, which means you have live microbes. that you can detect, or you can say there was at least the presence of these microbes in the sample, but maybe we don't know how to culture them, or maybe they just didn't do well in culture. There's definitely the list of microbes that we don't know how to culture, especially now that we can really detect them by their genetic presence, is much larger than the list of microbes we do know how to culture. So we have the swab, and the swab has some microbes that grow on it. There was something at the factory, or something lives in the machine that makes these or whatever, and it just gets a little bit of microbes on the swab, because it's actually pretty hard to get anything that's truly free of microbes, and it's even hard to get something that's truly free of microbe DNA. Oh, and Sarah raised her hand. Is that for a question?
Oh, no, that was for the attendance. Sorry, I forgot. I was just putting it down.
Oh, okay, cool. All right, so anyway, so you can have microbes living on your swabs. You can also have microbes living on your beads, and I can tell you that the ceramics or whatever other material suppliers or the glass suppliers that supply beads, they don't necessarily clean those beads. And a lot of times those beads can actually come in even dirty. Sometimes they come in just like even visibly with like crud on them. So it's also not just a question of things like beads or the sintered glass used in the extraction filters that people run their DNA through, basically the silica matrices. Those can come in also with their own biofilms on them. Those can also come in dirty. And this is something that's not completely appreciated. And for some samples, it's not actually even an issue. And this will kind of give you an idea of how it happens and can be an issue. So I can't really distinguish between microbes that were on the swab versus microbes that were in my sample. Now, if what I'm swabbing is a stool sample, stool tends to be extremely high microbial load. Stool's, extremely high bioburden or microbial load. And these microbes are going to completely overwhelm the signal I generate from just a sparse couple microbes in here. And they may be even completely different. So especially if I do a lot of characterization, well, you need to do a sample that is just testing for basically bioburden, which is to say that you run something through with basically nothing on it, just sort of exposure to air. And I can probably detect these and I can separate them from sort of the stool microbiome, but realistically, these are going to be such a tiny contributor, they're just going to show up as something extremely rare and probably not important. If I do, on the other hand, a tear sample, and I've actually seen people testing things like infant tears. Tears have extremely low microbial load, especially infant tears. Tears have extremely low loads of microbes. And at that point, as you can see here, I would, for example, get 3 microbe signals from the swab itself, and I'd only get so basically 1/4 of my signal would be from tears. So I could very easily overwhelm the signal in the tears with signal from just a dirty swab. And this is actually an issue that comes up a fair amount, especially if you're testing things with sort of low biomass of bacteria. So for example, people testing air samples, this becomes a huge issue. People testing places that should be relatively clean, such as cerebrospinal fluid, which generally should have no microbes in it whatsoever. That becomes a huge problem because then you're going to start detecting everything else. And actually, even certain places on the skin tend to be, they have known microbes that are expected to be there, but they actually have fairly low microbial load. So this is something to keep in mind, is try and have a good handle on what microbes might be present in your sample handling pipeline. And another thing to remember is microbiome is a living sample responsive to environmental conditions. And what I mean by that is we know that microbes like different environments. And this was actually an issue that the American Gut Project had fairly early on. So this is looking at stool samples at room temperature. I can't say I don't know for sure what any of these taxa are that are indicated here, but as a sample set out for a day, some new taxa started to appear. And this, I can tell you, is almost certainly proteobacteria and probably E. coli. So it turns out that despite that everybody's familiar with E. coli and thinks of it as kind of like the quintessential stool bacteria, E. coli is actually a fairly minor player in a stool sample. However, E. coli is a facultative. E. coli basically likes oxygen. And this actually becomes a big issue. E. coli does reasonably well at low temperature. It'll still continue to grow. And it not only tolerates oxygen, but it likes oxygen. And this ends up becoming a huge issue. So these samples were mailed in without any refrigeration or preservative. And what happens is a stool sample inside the patient is at about 37 degrees centigrade, and it is in a low oxygen environment. Once that sample leaves the patient and goes out into the atmosphere, it's now dropping to 20-something degrees probably, or whatever the ambient room temperature happens to be, and it's now exposed to oxygen. Those microbes are not all dead. In fact, those microbes should be coming out pretty much alive. And this is where things start to get really annoying, because stuff like E. coli that tolerates the cold and likes the oxygen is going to start growing. There's also a lot of microbes in stool that don't tolerate the oxygen. In fact, when you expose them to oxygen, they either stop growing or they even start dying. And they also don't tolerate the cold. So if you put them at below body temperature, they just stop growing entirely or even start dying. As they start dying, E. coli and other things that start growing are going to eat them. And that's basically what you see here. And I believe this actually really hurt their paper. I was told by somebody that this actually got their paper into American Society for Microbiology instead of being like something along the lines of like Science, Nature, or Cell or whatever. Although that's just a rumor I heard. But it was painful for me to see this, because I get why they did it, and I think they were probably right to do it so they didn't have to get rid of a really big and otherwise potentially useful data set. But they ended up having to bioinformatically remove E. coli from stool samples, which obviously is not something you want to do. It's also been found that the use of RNA preservative can cause a loss of bacteroidetes. This is not all RNA preservatives. There's different RNA preservatives out there. And in a few minutes, I'll show you why this happens, and also what you need to look for in an RNA preservative to maintain your sample and maintain its integrity. But we see here something that's interesting. We actually have a loss of bacteroidetes. And typically, as I showed before, bacteroidetes are gram-negative, so usually bacteroidetes tend to be overrepresented. In this case, they're being underrepresented. We can also cause a loss of bacteroidetes through freeze-thaw. So we see here, this is stool without preservative. And just by putting it through 5 freeze-thaw cycles, bacteroidetes have essentially disappeared. And this, we were able to figure out pretty quickly what's going on here. You have gram-positive microbes and gram-negative microbes. Again, gram-positives are tough. Gram-negatives are easy to lyse. So when you freeze water and it forms ice crystals, water expands as it freezes, which means you're going to have water inside the microbes that also expands as it freezes. And anybody who's lived in a place that gets cold probably is familiar with the idea that you need to make sure your pipes don't freeze. Because if you have a pipe that freezes, that pipe can sometimes just kind of rupture on you. And that gets really bad when the water unfreezes and all of a sudden you have a burst pipe now spraying water. So you freeze them, you're going to get a lot more rupture in the gram-negatives. And that kind of makes intuitive sense, because obviously if your pipe is thinner or your pipe is more flimsy, it's more prone to rupture versus the harder gram-positives that have tougher cell walls. They can resist a little bit longer the pressure that's generated by an ice crystal forming inside the cell. So they'll also get ruptured, but they're going to rupture at a much lower rate. And these bacteria contain a couple things that we really care about right now. We really care about the bacterial genome, because that's obviously what we're monitoring and what we're testing for. That's our kind of primary analyte. They also, however, contain DNA degradative enzymes, so endo and exonucleases that basically just want to chew up this DNA. Now, the good thing is no bacterial species is going to contain a nuclease that is going to chew up its own genome, because that's pretty much an evolutionary dead end. If you chew up your own genome, you die. But a lot of these guys do contain enzymes that'll either chew up their own genomes if their genome gets fragmented. So those are exonucleases that basically start eating up broken DNA. They also contain endonucleases that won't cut their own genome, but will cut the genome of either other microbes or of bacteriophages. And a lot of these endonucleases are designed to basically interfere with bacteriophage development because they'll chop the bacteriophage genome up. Sorry, by day 3 of class, my voice always starts to give out on me unless I make sure I drink regularly. So anyway, we have our hard to lyse. So we have our hard to lyse gram positives. We had a little bit of lysis. We had our gram negatives, which we had a much more lysis. When they rupture, they're all going to spill out all these enzymes in DNA. The nice thing is freezing is also going to inhibit these enzymes, and these enzymes are stuck in basically ice cubes. When we thaw the sample, so we start applying some heat to thaw it out, these enzymes unfreeze. And now all of a sudden these enzymes are just basically floating free, and they're seeing a lot of DNA substrate around they can work on. And these enzymes just chew up a lot of that DNA. And so stuff that ruptured tends to be lost. So this led to the question of, will rapid denaturing or other inhibition of nucleases help? And it kind of makes sense that it would. So let's say that we have these things frozen, but we thaw it directly into some kind of an inhibitor or a stabilizer, so that as these enzymes start to thaw, they're also immediately hit with an inhibitor. Now all of a sudden the enzymes can't break down the DNA and the DNA is preserved. So that's basically how you deal with freeze thaw. But as I mentioned before, one of the inhibitors or one of the stabilizers, which is sort of an RNA preservative in this case, was actually showing itself to cause bias. So are they all suitable for this? And as I mentioned before, an RNA preservative, I'm not going to specifically mention it by name here. It was causing an overrepresentation of Bacteroidetes in the sample relative to fresh. I'm sorry, it was causing underrepresentation of Bacteroidetes relative to fresh, not overrepresentation. And this is basically what the issue seems to be, is if you freeze the sample and you have bacterial lysis, or if you just have bacterial lysis happening in the stabilizer itself, that DNA is going to wind up basically going into solution. And if the inhibitor can't go through the extraction process, so for example, RNA-later is a popular one. It's actually a very good nuclease inhibitor for RNA preservation. I've used it myself before for preserving samples and that kind of thing. However, one thing I learned very early on was you have to make sure you thoroughly wash it off the sample, because even a little bit of RNA-later carryover, because it has some kind of strange salts in it, can really interfere with DNA binding to a column, and it can completely crap up your extraction process. The problem with that when you're doing a microbiome is if you have all this lysis happening and you have differential lysis, and RNA-later itself is also fairly lytic for bacteria. It'll pretty much kill everything, and it'll lyse especially gram negatives. That DNA goes into the solution, and even though the DNases are inhibited, you have to spin down the cells to wash off the inhibitor. And when you wash off the inhibitor, you're actually washing off also the DNA that went into solution there. So it's kind of a complicated issue, but it'll make a little bit of sense it makes more sense when you think through it. Is just, you have to make sure that basically this inhibitor can also go through your extraction process harmoniously. So it's not going to mess up your DNA binding to columns or whatever else you're doing for extraction. That you can extract DNA without having to throw out inhibitor. Because if you have to throw out your inhibitor, then you wind up throwing out anything that went into the inhibitor solution. Adding sample direct to an inhibitor is about the same issue, because the inhibitor itself tends to be lytic, and obviously some things lyse easier than others. Okay, and then the last thing I'm going to go through, one of the last things I'm going to go through is microbial sequence databases. So Vivian's asking, does a preservative need to be washed off as well? And the answer to that is it depends on the preservative. So just as an example, RNA-later, that will tend to interfere with binding. I know, because I'm very obviously familiar with it, is Zymo has DNA RNA shield. DNA RNA shield is actually designed specifically to go through all of our columns without causing interference. So that one doesn't need to be washed off, which actually frees you from that whole issue of stuff lysing. Because I can tell you that DNA RNA shield is actually very lytic. It's actually a really nice way of destroying pathogens as well. I know some people who are using it for coronavirus sampling right now, which is actually what a lot of it's going towards at the moment. But it depends entirely on the preservative. Some preservatives have materials that are just incompatible with good DNA extraction, and other preservatives, and I know that we designed DNA RNA shield that way, the other ones are designed to be harmonious with extraction. And I know there's other preservatives out there, and I don't know enough of their specifics to be able to say what their compatibility is with applications. So does that answer your question? Okay, cool. Yeah, it's just a matter of basically knowing the documentation on your preservative and checking its compatibilities with different processes. But I just happen to know that RNA later is one that tends to be very incompatible because it has some strange salts in it. All right, so microbial sequencing databases are kind of an interesting one that a lot of people don't give thought to. Typically, if I say things like, you know, there's the Silva database, there's the Green Genes database. Some people may or may not know what the differences are between them or what the ideas are behind them. So I'm just going to go through a very brief history of microbiology and especially how it was funded. And that'll help us kind of understand why our databases are the way they are. So initially, there was kind of a big explosion in Dutch science, and a guy named Van Leeuwenhoek, he didn't invent the microscope as we know it. Actually, our modern compound microscopes are very different than what he was using. He came up with a really good way of basically making these highly polished glass beads. And they make kind of these annoying microscopes that look more like a kind of a square tile with something in the middle of it. He wasn't trained as a scientist. He was actually trained, I think, as a tailor and a businessman, and I think he was also a local politician or something. But he was a very curious individual, and he kind of had the mind of a scientist. So he actually just went using his microscope to look at everything and discovered that pretty much everywhere he looked, he could see these things he called animalcules or animalcules or whatever. And what he was describing were basically the first time a human ever realized that there's bacteria in the environment. And so what's interesting is he basically became the original microbiologist, and the original microbiologist was an environmental microbiologist. He just realized that, you know, he could find these things in rainwater drops and pretty much anything else he looked at. He looked at plaque between his teeth, and that was just teeming with bacteria. He also invented the first antimicrobial, which was vinegar, because he actually was observing that the bacteria from the plaque in his teeth were moving around a lot. And then he said as soon as he added a drop of vinegar to them, they all stopped moving and basically went dead. So he actually invented also the first antiseptic. Then this guy named, and this was mostly curiosity for I think about a century. Then Louis Pasteur came along and realized that, so he was actually first paid by, I think, the French wine. He started off as a chemist who was trying to figure out, trying to understand different wine compositions and why wines taste different. And he was commissioned by the wine industry, which was one of the biggest industries at the time in France. And he started figuring out, during this process, he was trying to figure out how wine ferments and also how wine spoils. And he figured out that wine fermentation is a microbial process. There's basically microbes that do the thing. He also figured out that wine and other food spoilages is also a microbial process. And then he and Robert Koch also figured out that microbes are making people sick. Prior to this, they thought it was either like the bad air given off by a dead person or a dying person. So it was called the miasma theory. Some people thought it was just evil spirits or vengeful deities causing illness. But he and Koch were able to prove that a lot of illnesses are actually caused by microbes. And of course, this made microbiology the in thing. because basically we figured out that microbiology is important commercially for some of the biggest industries. It's also important because it's taking a lot of lives. And then in the later half of the 20th century, biochemists and molecular biologists tried to understand basically how life works. And E. coli provided a really simple basic model for doing this that was shareable between labs, grew up quickly. And as far as a life form goes, it's a fairly simple one. So what are the consequences of this? Well, van Leeuwenhoek did his work, but it kind of remained a curiosity. So microbial ecology was actually the first form of microbiology, but the interest in it was limited because nobody was actually seeing money to be made off of it. Pasteur figured out that microbes are both killing people and making and destroying our food. Suddenly, microbiology became of big general importance because it was having huge health implications and people were dying left and right of infections. A lot of these infections are now very easily treated with antibiotics. Microbiology also became a great financial interest because a ton of our products turned out to be commercial products, and a ton of our products that were basically spoiling on the shelf. It turns out, and this is where pasteurization comes from, if you heat them up and seal them so that the microbes inside die and new microbes can't get in, they either spoil much more slowly or they don't spoil at all. And then finally, biochemists using E. coli as kind of the workhorse of the lab meant that everybody had a bunch of E. coli strains sitting around that had some different properties. They were all super well characterized, and everybody wanted their favorite E. coli strain sequenced. And obviously, this was a big deal because molecular biology was well funded, and a lot of people realized that understanding the basic mechanics of life was really a key to scientific advancement. So this brings us to our databases and why our databases are good for some and terrible for others. So you end up getting what you and the others pay for. The others often being the NIH actually, which I guess is funded through us. So if you find E. coli in your sample, I got good news for you. You're going to be able to compare it to like the 100 closest strains of E. coli that are in the database. E. coli are very highly represented in databases. If you find a pathogen or something that's very similar to a pathogen, it's going to be well represented in the database because, not surprisingly, one of the best funded microbiology endeavors, and in fact, some of the only funded microbiology endeavors early on, were looking at microbial pathogenesis and trying to figure out which bacteria are causing diseases. So you're going to find at least one version of whatever it is you're looking at, and you might even find a few strains to compare to. If you're looking at microbes that live on humans or animals that are similar to humans or just common animals, these are also typically pretty well studied. People figured out there's commensal bacteria and even kind of symbiotic or beneficial bacteria. People figured that out fairly early on. So these are reasonably well studied as well. If you're studying food or fermentation or microbes that are potentially going to show up in those contexts, your microbes are also going to be really well represented because there's a lot of money for commercially important microbes. And this is going to be paid for even by some of the big food manufacturers. And then finally, if you're studying environmental microbiology, especially in the more remote or obscure environments, so if you're doing microbial ecology of built environments, that's improving a lot, and there's stuff being added to databases all the time. If, however, you're doing microbial ecology of an underwater cave, an underwater cave in kind of a hard to reach place, you're going to have, especially if this cave has some unusual properties that are, like this cave happens to be very acidic, you're going to wind up finding that your microbes aren't represented in databases. And actually, even worse than that, you're probably going to also wind up finding that if you try and use a database for comparison, the database may actually start pointing in the wrong directions because you may have some programs that are finding you spurious connections to microbes that are unrelated to what you're into just because completely different environments such as the human are much more studied. So this is something to keep in mind with environmental microbiology is if your environment is not super well characterized, the database, you're going to be putting more into the database for a while than you're going to be getting out of it. And this brings me to my next point, which is how stuff gets into databases. So researchers are really strongly encouraged to submit new sequences to databases. You pretty much have to do it to publish. Also, a lot of people just sort of mark down as kind of a mark of their productivity in microbial ecology, how many new sequences they've contributed to the databases. They're sometimes not always as strongly encouraged to have rigorous quality controls. So stuff that I've seen in databases includes stuff that is obviously a chimera. Basically, somebody didn't do a good chimera removal process. I found cases where I have the same exact sequence listed for more than one species. And in fact, if I trace it back, I can pretty much figure out that somebody just made a boo-boo when they were uploading the sequence, and they gave it the wrong name. Or I'm sorry, in this case, I found so this one I haven't done myself, but I know people who do taxonomy trees on databases where they can show there's a branch that just kind of goes across different groups, where it was clear that somebody uploaded something as, a B subtilis strain that they discovered. But if you look at the taxonomy, it's clear that they either, that they had the thing named wrong, and it was probably some other species entirely, and they just gave it the wrong name, because mistakes happen. Sequences can also be found that are identical between poorly related species, and the same idea as above. Somebody just made a mistake on the name. They had an Excel sheet and a line got shifted or something. Another thing I've seen in some databases, although not so much microbial databases, is there's a really well-known gene called OCT4. And if somebody is moving a table through Microsoft Excel really likes to change OCT4 to October 4th. And I've seen a few databases that actually have gene sequences called October 4th. And I was like, somebody put this thing through Microsoft Excel. And that's kind of a notorious problem. And somebody actually did a study where they showed how many databases show evidence of Microsoft Excel contamination. So a lot of times what you'll see is when databases get updated, they don't just contain a significant amount of new microbial material, because a lot of databases are now doing curation, which is where they look for stuff that looks like it's probably garbage, and they remove it. So you'll see a lot of stuff that gets submitted doesn't get approved because it's either poorly documented or there's some demonstrable error with it. You'll also see that with database updates, something that was there previously will disappear because somebody found evidence that it was garbage. And most importantly, especially if you're doing microbial ecology and you're going into these new environments, don't contribute garbage into the databases. Make sure that you have good controls. Make sure that your bioinformatics pipeline is rock solid and automated, and that you're checking for any of these potential issues. Because obviously submitting garbage to databases not only hurts you, it hurts a lot of people down the line. And if a lot of stuff you contributed has to be removed, that actually messes with a lot of people's experiments in the future. All right, and the last thing I'm going to go into very quickly is diversity metrics, because this comes up as probably the most common thing that's measured on microbiomes. So alpha diversity, this is actually my favorite measurement. And this is actually one of my favorite things to look at with a microbiome. So I sometimes look at microbiomes in context of infection. And one of the first things that I'll frequently see is the alpha diversity goes way down, and you get one predominant species, and that predominant species is often a known pathogen that's causing the disease. So alpha diversity is asking the question, how many different species are in my sample and how evenly are they distributed? So just as an example of this, a sample that's a monoculture, think along the lines of E. coli that you're using to grow up a plasmid in the lab. So it's basically just a culture of pure E. coli of one strain. There's no alpha diversity there. There's no diversity in there whatsoever. It's a monoculture. So this is the lowest possible point for diversity. You have one microbe and it's the whole sample pretty much by definition. If you have a sample where you have multiple microbial species present, but you only have one species that's super predominant, that's also going to give you a pretty low alpha diversity. And obviously a sample where you have very few microbial species present is also going to tend to give you lower alpha diversity. Alpha diversity is an intrinsic property of the sample. And I say this because I've seen a lot of people, especially recently, trying to use alpha diversity as a surrogate for how good their their microbial prep was, or how good their bioinformatics pipeline was. Excuse me for 1 second. And as far as I'm concerned, this is this is essentially a mistake and not a valid measurement. The alpha diversity of a sample is what the alpha diversity of the sample is. A human stool sample can be expected to have much higher alpha diversity than, say, the vaginal microbiome. The vaginal microbiome is kind of known for being a very low alpha diversity sample, where basically you're just looking at a lot of different lactobacillus strains, especially Lactobacillus crispatus, but a few others as well. So if you measure a vaginal microbiome and you find an extremely high alpha diversity, I've seen some people claim that getting a high alpha diversity is a sign of a better extraction. But if you get a high alpha diversity on a sample that is just inherently low alpha diversity, then you probably screwed up somewhere and you're detecting garbage. Alternatively, people operate under the assumption that stool should have high alpha diversity, but in fact, the alpha diversity is just a property of the stool sample. Different stool samples would be expected to have different alpha diversity. And another scenario that comes up a lot is, I kind of showed earlier how firmicutes can be underrepresented due to poor lysis. So if I have a sample that has a large number of gram-negative species in it, but is almost entirely, let's say, 80% firmicutes, it should have low alpha diversity because you have basically predominant firmicute. If I were to underlyse that firmicute, I could actually see the alpha diversity improve just by the fact that I had a predominant species and I made it less predominant due to technical issues. I just underlysed it and didn't pick it up. And now all of a sudden, the less predominant species become more predominant, which makes my alpha diversity look better. So alpha diversity is a property of your sample. I really get annoyed when people treat it as alpha diversity is also a marker for having done something right or done something wrong. Beta diversity, that's actually comparing two different samples and how similar or different they are. So the gut and the oral microbiome tend to have fairly high alpha diversity. There's a lot of different bacteria that are pretty well mixed. But if you take two oral microbiomes, for example, they're going to tend to have low beta diversity, especially if you're dealing with similar individuals, because even though they have high alpha diversity, the two samples are actually fairly similar to one another. Another example of this is during irritable bowel disease, and I think this is pretty well known. There's massive changes in the gut microbiome. So I think the comparison I've seen, and this was actually, this was done by the Rob Knight lab, where they were just tracking an individual with irritable bowel disease during flare-ups and during healthy periods. And what they determined was during a serious flare-up, this individual's gut microbiome wasn't slightly changed. It was actually the difference between, say, for example, the microbiome you'd expect in a desert environment and the microbiome you'd expect in a sea environment. And you can quantify those changes by looking at, among other things, the beta diversity between samples. So the beta diversity between his sample healthy and his sample during a flare-up, or even other humans and him during a flare-up, was bigger than the difference between basically the desert and the sea. And that's essentially what beta diversity is. You may hear people talk about gamma diversity. Gamma diversity isn't usually a big issue in microbiome analysis. I've never seen anybody actually use it besides saying gamma diversity also exists. It's more interesting for sort of the more traditional ecology, but that's basically looking at a multi-site ecosystem. So within a site, you take alpha diversity. Between sites in the same ecosystem, or just between sites in general, you can take a beta diversity. And if you want to compare, say, for example, we're looking here at the side of a mountain. So if you want to compare the side of this mountain versus the side of another mountain where you have multiple sites, that's where you take gamma diversity. All right, and that is actually the end of the class. So I will take any questions anybody might have. Like I said, the final version of the slides will be going up in the next day or two when I get them polished up and I get the course kind of laid out so that it's more even. Because I'm aiming to get everybody out roughly at about 2 to 2.5 hours every day. So I need to even out the material a little bit, and I need to also put up the corrected pipeline slides. But I will stick around for a few minutes to answer any questions anybody might have. Otherwise, feel free to email me. I'm also fairly easy to add on LinkedIn, and I'm always happy to add former students on there and keep in touch. And the last thing is, if anybody has just microbiome related questions, I'm obviously more than happy to help outside of class. The nice thing is that Zymo, I work on microbiome, so pretty much any, and Zymo does have a big emphasis on tech support. So pretty much any time anybody has a question for me on microbiome, I'm more than happy to help answer it. And often, especially if you're in LA, which I know a lot of you are, it's very easy for me to just make a trip up there during the day. If you just want somebody to speak to your lab about some of these issues I talk about. You know, obviously right now I'm not going up to LA, but I can do a Zoom meeting. And I'm obviously always more than happy to show up once the lockdown is lifted. So I'll answer any questions anybody might have. And I think you guys should all have my email address. I think for the class, I typically use my UCLA one, but I basically answer them all. So Eva wants to know where the slides are being uploaded. So I sent out links to the sort of draft version of the slides. So you have my draft slides already in PowerPoint. There's going to be PDFs of my slides, and those are going to be uploaded to the course website. So the link I sent out last night to the course webpage, that still has the old slides on it. Those are going to be replaced with my new slides. If you look through the old slides, you'll understand why I changed the course around so much over the last, for this round. I don't know if anybody out there is even familiar with 454 sequencing, but the course had a lot of material in it that just wasn't current. The other thing is also my email address again. If you, please feel free to email if you have feedback on the class. If there was some topic I did not cover that you wish I had covered, or if there was a topic that I covered that you thought, that I thought was unclear or unrelatable, please let me know, because as I said, I'm polishing out my slides over the next couple of days, and I can kind of incorporate that feedback, and it's kind of the perfect time to do it because I'm building out the course. I'll send out my email. I think everybody got it last night also, but all right. And everyone, you're welcome. It was a pleasure teaching. Thank you for being my first class for this. It was a lot of fun making this course. Obviously, it was a fair bit of work, but I think it's turning into a good product. And as I mentioned, I spoke to some of the people at Zymo who do the marketing and making the educational videos. And I'm going to have an educational video out probably next week at some point. And it's going to be on the ASV versus the OTU method, because I think that's actually an underappreciated area in microbiome, especially for people doing targeted sequencing. All right, so if there are no questions, oh. I have somebody telling me slide 13 from yesterday's slides was not readable. Okay, I will look into slide 13 from yesterday and figure out why it's not readable. All right. So I think if all the questions are solved, I'm going to terminate the meeting, and this will be on video. This should go up on video in the next few hours. I think I'll send it to Eli to post.